I'll just do a quick introduction. Jim is at UN. He's a professor in the Department of Biological Sciences at UNH. He's the director of the UNH Center for Freshwater Biology and co-founder of the New Hampshire Lakes Lay Monitoring Program. He teaches lots of classes, great classes focused on ecology, lake ecology, general ecology, stream ecology, and does his current research focuses on the transfer of cyanobacteria toxins within lake food webs. So cyanobacteria, of course, something that we're concerned about here in Maine. So thanks so much, Dr. Haney, for joining us. And folks who are watching, if you have questions at any time, use the Q&A function or the chat function. It's also always helpful to know who's on the call. If you want to introduce yourself by chat with your name and where you're from, put questions in the Q&A box, and then when Jim's finished, we will open it up to questions. So with that, take it away, Jim. Thanks so much.
Thanks very much, Susan. It's a great pleasure to be here. I have always admired the work that's being done on lakes in Maine. You have to excuse me, I have a little bit of laryngitis today. But both from the state, but also the citizen science in the state of Maine has always been so impressive that it's really a great pleasure just to talk to you about some ideas that we're working with at the University of New Hampshire. And so I'm going to begin by talking, first of all, about a revelation I had a few years ago. Many of you probably had it when you're in high school, but I used to think that science was sort of the search for answers. And then I guess after conducting science for so long, I began to realize that it was really the questions that were the most exciting parts of the science. And so the question is, I guess I have, is what controls when the questions are best asked? I think one of the things, of course, is time. Timing is very important as to when you ask a question, but it's also relative to the technology and the information that's available. So the questions we had 20 years ago or 30 years ago are not the questions we're asking today. On the other hand, we have questions that we're not quite ready to answer, at least yet, as I would say. So let's back up a little bit. I refer to these as HCBs. The older literature and some of the expressions still calls them HABs, but basically harmful cyanobacteria blooms are easy to recognize. They're the scums that occur on the lakes, and they may be just a small filament or two, such as seen in the Old Durham Reservoir, or they could be entire lakes for kilometers. Actually, I developed my interest in cyanobacteria while on a research tour in New Zealand during the sabbatic back in 1987. At that time, toxins weren't very well known, and they were concerned because some of their lakes would develop blooms that would last for even a year or two. So the question was, what's fueling that? And through that, I began taking an interest in cyanobacteria. So one of the questions, because we talk about blooms so much, is how serious is the problem of cyanobacteria blooms? And one of the things that, of course, makes us aware of the potential problems is the deaths of pets, dogs in particular, that every year occur all over the country and in other countries as well. And so that dogs, because they're certainly not afraid to drink water that's heavily polluted, often then are the ones that die first around the lake. So I'm not going to talk about toxins much, just to mention that cyanobacteria, as most of you would already know, produce a wide range of toxins. Some of them are referred to as liver toxins or hepatotoxins. These would be microcystins, cylindrospomopsin and nodularans, although it's now known that these liver toxins also can affect the nervous system and the digestive system and the muscular system. Neurotoxins are also produced, and these include the anatoxins, the neosaxitoxins, and the BMAA. The BMAA is the one we've begun working with because of its connection with Alzheimer's and ALS, but we're also working with microcystins and anatoxins. Excuse me. So what are the links to human health? I mean, how serious is the problem? Ultimately, we want to know, is it affecting human health? And we began our interest in this back in 2007, published a paper in 2009 on the fact that there were high densities of ALS patients around Mascoma Lake. It was no particular reason to believe Mascoma Lake was the cause, but we began looking at that as a possible source. Since that time, there have been several epidemiological studies done, and these are actually even more interesting. One is the effect of liver disease, non-alcoholic liver disease, and water quality. And Zhang in 2015 was able to show there was a positive correlation between these two factors. And then later, actually, Torvik, a colleague of mine actually, began looking at the epidemiological areas of New England and ALS and found that the likelihood of belonging to a cluster of ALS around a lake increased with the decreasing water quality and the increase in cyanobacteria. One of the interesting things is that this statistical influence, I should say, seemed to extend from 10 to 20 or even 30 kilometers out from the water source. So the question is there a link? Is there an environmental factor here involved? And could it be airborne? So one of the questions that we have, of course, is, what we see are the blooms. And are these really the problem? And this is a gliotrichia I put on here, in particular because Maine has so many gliotrichia blooms or gliotrichia in their lakes, in fact, a very common cyanobacterium. And this brings up the bigger question, which is, we see blooms, we understand what they are very easily because we can see them. But is it really what we think we're looking at? We all know, of course, that we see a flower, but what we see is not the flower that a pollinator sees. And in that sense, what we see is not the reality of all organisms. We also know that just because we don't see something doesn't mean it can't harm you. And we're most aware of that because of the airborne viruses that we're now contending with. So what are blooms then? Well, blooms in a very, very coarse sense are evidence that cyanobacteria are in the lake. In a poor analogy, I would say, they're sort of similar to poxes, because you know that there's something wrong when you see pox on a person. It could be measles, it could be smallpox, it could be chicken pox, but it doesn't really tell you too much about the actual underlying condition. And in a sense, what we see on the surface of the lakes doesn't really tell us a great deal about what's going on within the lake. So I would simply say that since HCBs can be very toxic, they obviously are important. But the cyanobacteria as a problem is more complex than just the surface blooms. So let's start off by what we think of as the cyanobacteria in the lake, as in a very simplified form, could be looked at as two types. the large types, which are what we call the BFCs, or bloom-forming cyanobacteria. They form highly toxic scums on the surface of the lake. And that's what we see then as HCBs or HABs. The picocyanobacteria is the other group, and these are what we think of as the invisibles, just the opposite. They're so small that they're often overlooked with a light microscope and require fluorescence, such as shown in this picture, to illuminate and to visualize. Their importance is that they feed into the food web because of their size, and they also can enter the air as lake aerosols. So if we look at the lake now and think of these two groups, what we would see then is the group of picocyanobacteria, the small, less than 2 micron size, are what we call edibles, mainly because they're edible by the zooplankton. And as such, they can be transferred up the food web and eventually get to the top consumer, which could be, in this case, the common loon. And so this would be then the movement of cyanobacteria and their toxins through a classic food chain. The BFCs that we see on the surface don't appear to contribute a great deal to the food chain. Instead, what happens to them very often, and what we see, although not always, is that they can move to the surface given the right conditions and form then a surface bloom, which we then recognize as this typical scum. So this transfer up the food web turns out to be quite important. The zooplankton, such as this Daphnia shown here in the movie section, which was from Lake Attitash, just over the border in Massachusetts from us. If you watch it, you'll see that the large particles, this one just went in, just got kicked out again. That's a microcystis type, actually Vornlichinia. And here is a filamentous form, which is Dolichospermum. And it can't be eaten either because of the design of the filtering apparatus. It's just too large, so it gets picked out. As a result then, these large forms often prosper even when there are zooplankton present because they can't be eaten. So do these toxins accumulate in the food web? One of our earliest attempts at looking at this was in Province Lake in New Hampshire and Maine on the border, over in Wakefield. And we measured then microcystins in some of the components of the food web, in the phytoplankton, in the zooplankton shown here as the Daphnia, in the largemouth bass, in the pickerels, and also in a mussel. And what you see is clear evidence that these toxins do become incorporated into different trophic levels in the lake food web. And therefore, we think of this as bioaccumulation in one sense at least. But these are microcystins, liver toxins. Notice that, in fact, in the pickerel, the largest amounts of these liver toxins were in the liver, as you might expect, and also in the spleen, which other studies have found as well. So it does accumulate. One of the questions is, does it go to this top level we talk about, the top consumer? And we had a chance to check this by a collaboration we did with the Biodiversity Research Institute in Portland. It was collecting loon blood at the time, and we collaborated with them, outfitted them with limnological equipment to collect plankton and toxin samples from the lakes. And we compared then essentially what took place at the first trophic level exchange between the phytoplankton and the zooplankton as a ratio. In this case, it was with the neurotoxin BMAA. And then how much BMAA was in the blood of the loons. And one of the interesting things that we found is that there was a significant relationship between the ratio, how efficiently it was transferred from the zooplankton, or from the phytoplankton to the zooplankton, and how much was in the loon blood, which supported the idea that in fact these toxins are moving up the food chain or up the food web. So one of the questions is, are we already monitoring cyanobacteria? Are we already monitoring HABs or HCBs or CHABs, depending on the acronym you like? So that's a good question. And before I answer that, or try to answer that, I'd like to simply take you on a review of what we've been doing with lake monitoring and what questions we've been asking. So the early questions back in the 1940s and then later the 50s and 60s, eutrophication became a big issue. And the question that was being asked by monitors at that time was, are the lake's trophic status changing? Is a particular lake getting better or worse? And since that hadn't been done, we had no long-term information. This was very new and exciting at the time. So to do that, as most of you would already know, the tools that were being used were Secchi disc to measure transparency, water samples for phosphorus, and also water samples for chlorophyll. So Secchi disc, phosphorus, and chlorophyll were the three major parameters that were being used all over this country and other parts of the world, in fact, to ask the same question. So it's interesting that beginning in the 1990s, I would say, roughly, and for the past 2 decades, there's been a growing interest in cyanobacteria, and especially the toxic cyanobacteria, and the health issues that they have for pets and for humans, and also for wildlife. So the monitoring then is having to adapt to a new set of questions, which are different in some regards to the old questions. That is, the questions today seem to me to be more immediate. Rather than looking at long-term trends, some of the questions are simply, is the water safe? Is it safe to drink? Is it safe to recreate in? Is the air around the water safe to breathe? So these are questions, because of the seriousness of the problem, no longer are we satisfied to simply look for long-term trends. The questions really are more, I need to know now. And then the other questions are, how do we control or prevent, or if you're especially in the business of drinking water, how do you predict blooms? So these are questions that the old monitoring didn't really tackle head on. So new tools, new approaches are clearly needed at this time. So if we go back in time, this is again about 10 years old, this picture on the left, showing a very, very cold undergraduate assistant who's operating one of our original plankton separation devices. Because at that time, we wanted to be able to analyze the plankton in different fractions. We didn't want to mix everything together. So we developed this technique of darkening these separation bottles so that the upper region then would be dark and without wave action, so that when we put lake water into it, the bloom-forming cyanobacteria over time would rise to the surface. And then in the bottom part, which was clear, the lighted region, the zooplankton, much like insects, are attracted then to bright light, and beam light, that is, and they then move down into the tube, where they then can be filtered off onto a Nitex filter. Shown here on the right is an example of one of our lakes, which is dominated by Daphne, as you can see. So, and by that means, you can then remove the BFCs, or bloom-forming cyanobacteria, by collecting just at the surface. And then you also have a sample of the zooplankton. So now if you want to ask questions about transfer of toxins, you can. So this was, however, never thought to be the last version of this particular device. It often would fall over in the canoe and we'd have to start over again. So Nancy Leland and I, some years ago, began working on a new and improved mousetrap, so to speak. So we said, surely this can be improved upon. So what we did, it resulted in what we call now the zapper, the zooplankton and phytoplankton photoresponse mechanism, because you pour the water in and the dark bottle then allows the cyanobacteria and other phytoplankton to stay at the top. The lighted region here, and you can attach a light to it, in fact, to speed up the process, removes the zooplankton. And so this zapper now is also in a form that one can buy, actually. This is called the pocket zapper. So this is something you wouldn't want to be without, so you keep it in your pocket, obviously. So the accessory pigments are an important part of the approach that we're taking now to cyanobacteria monitoring. In addition to chlorophyll, which is the pigment that has been monitored for, again, almost a half century at least, we're adding two other pigments. One is phycocyanin, which is a blue pigment characteristic of most of the cyanobacteria and very few other groups, although there are some exceptions. And then we've added to it phycoerythrin, which is a red pigment. And this then is especially found in the picocyanobacteria, but also in some of the other cyanobacteria. So now these accessory pigments, which cyanobacteria evolved over millions or more years with the purpose of collecting more energy. So chlorophyll collects energy primarily in the red and blue wavelengths. And now you can collect in other wavelengths, light orange, light yellow, and that energy then is funneled down into the chlorophyll molecule. So that's why it's an accessory pigment, so that it just collects extra energy and makes it then possible for cyanobacteria to sometimes exist whenever the light is too poor for other phytoplankton, and gives them a definite advantage. So the pigments are very important for a lot of reasons. They allow us to track cyanobacteria, and they sort of act as thumbprints for both identifying the cyanobacteria, but also for predicting the toxicity associated with them. So the fluorometers we use have essentially three lasers. One is the laser, the blue laser for chlorophyll. When that particular wavelength is absorbed by chlorophyll, it gets excited and gives off a signal that can be measured. And the chlorophyll then is actually identifies essentially all the phytoplankton. The phycocyanin absorbs light. It's a blue and it has a yellowish light that it absorbs. And phycoerythrin, again, a separate wavelength. And by measuring the relative amounts of these three pigments, you can get some idea of what types of cyanobacteria are present. For example, the chlorophyll, again, tells us simply that they have photosynthetic pigment because all of the photosynthesizing phytoplankton have chlorophyll. Phycocyanin, on the other hand, is more characteristic of the BFCs, such as microcystins, for example, and some of the Delechiospermum. And phycoerythrin, a large signal of phycoerythrin, can tell us that it's likely that Cyanobacteria are present in abundance. In addition, one of the things we've been discovering is that these pigments can also help us identify which are the toxins we should be looking for. For example, if we have a high phycoerythrin count, it's likely that BMAA is one of the dominant cyanotoxins. If phycocyanin is the dominant, very often it's microcystins or the anatoxins, the neurotoxin, anatoxin. So again, it's not 100%, but it gives us an idea of where to look, which is important. So why do we measure these pigments? Well, one of the reasons, of course, is that compared to testing for toxins, and depending on the method you use, it can be hundreds of dollars per test. Pigments can be relatively rapid. You can certainly do them in the same day. And if you're in a hurry, you can do them within hours or less. They're relatively simple to do the measurement of pigments using fluorometry. It's simple and minimal amount of training is needed. And perhaps one of the really important reasons is you can spend a lot of money testing for toxins, especially when it may or may not be necessary. Whereas with fluorometry, once you have purchased a fluorometer, the actual testing with it is just pennies. So what do you get for all this? Well, one of the things that we've discovered is that when we looked at the relationship between pigments and toxins, and this one here shows the relationship between phycocyanin, or PC, and the toxin microcystins, MC. If you just throw everything together, there's a lot of variability here. It doesn't look like much of a relationship. But if you actually identify what the dominant BFC cyanobacteria is, what you see is that if it's microcystis, which are the red dots here then, those microcystis-dominated lakes actually have a fairly close relationship to between the pigment and the toxin. And if you separate out the dolichospermum or anabina dominant lakes, they too then have much less variability, and you can then develop a relationship there that can make predictions. Where you have a mixed assemblage, it's somewhere in between, obviously. So identifying who's producing the toxin is quite important to establishing what the relationship is, because essentially microcystis produces lots of microcystins. That's what it's named for. The toxins are named for. The dolichospermum produces some microcystins, but much more, it produces anatoxins, for example. So that now we have to add some other information into the equation. So you can see then the benefit of knowing not only this relationship, but who the dominants are can help you then. So this is then allows us to make some sort of a first order estimate of how much toxin is present from the amount of pigment present. So one of the questions, and I said this would be empowering people to measure cyanobacteria. So how does it work? How do people become empowered from what we're doing? Well, that may be a little overstatement, but let's just describe what we're doing. So basically, our group, which is our Center for Freshwater Biology Laboratory, essentially establishes then some sort of relationship with citizen science groups. On the Cape, we're working with the Association for the Protection of Cape Cod, or APCC, and they are sampling lots of lakes. And recently, we're beginning a relationship with high schools as well. And then the relationship between those two, essentially, the citizen scientists collect the samples, do some of the processing, in some cases, surprising large amount of the processing, but the actual toxin testing and development of new techniques for monitoring is pretty much what we do, or what could be done by other, what I call here monitoring advocates. Could be lake associations such as your own, could be any group that then would like to take on that role. So what happens then after you collect the samples? Well, first of all, one of the ideal ways is you have a sample collection kit or something equivalent to it. This can be put together by anyone. The hose for the collection of water samples, for example, the microscope for identification, the actually the pocket zapper here for separation, and then one of the surprisingly least expensive piece of equipment, but most essential, is a small bottle with a piece of gauze attached, a 50 micron mesh attached, through which you pour the water sample. In doing that, you begin to reveal lots about what's going on in your lake that you would never learn from whole lake water samples. So the size fractionation into the less than 50 micron, and then the BFC isolation through the pocket zapper allow you to then identify the cyanobacteria either with the microscope or with the fluorometer. And the fluorometer then also gives you the pigment analysis, the amount of chlorophyll, phycocyanin, and phycoerythrin. That combined then with the toxin analysis is a powerful predictor of what's going on in your lake and can allow you to make some statements. So as I wanted to point out to you, and I forgot to do this before, let me just back up. Right here, is that one of the things about this equation then is that using this, you need to have the identification of the dominant forms. And there's a counting method we've now submitted an SOP for that's available, so that you can essentially learn this for any of the toxins that are produced and any of the pigments that you collect. So this then is a powerful tool because it allows you to say something about what you expect in the way of types of toxins, but also the amounts that are present. So let's talk a little bit about the goals of the cyanobacteria monitoring program. And I want to leave enough time that we have a chance to answer questions and talk about some of these ideas. So first of all, the goal of this cyanobacteria monitoring that we're doing, that, as I say, goes sort of beyond water quality monitoring, is to track the changes in the abundance and the composition of the cyanobacteria through pigments and identifications. Again, it's the, if you think of a lot of water quality monitoring is sort of the equivalent of taking the blood pressure, temperature of a patient. We need to get inside and see what's going on. How does that patient function? Turns out that lakes are very much like people. We're both ecosystems. Humans are ecosystems, lakes are ecosystems, and they have a lot of parts. So if you're going to find out how your lake is, why it's doing what it's doing, you have to know what are its characteristics. The second goal is to predict bloom problems in advance. This can be useful for a lot of reasons, as I say, for obviously, if you are using surface water for drinking water, that's very important, but also for recreational purposes, and also for essentially warning people ahead of time that there is going to be a problem. Cyanobacteria, what we can do with the pigment estimates is use pigments as a surrogate for biomass and develop growth models. This is all laid out in some papers we've written, and be happy to share that with you. But when the growth rate of the cyanobacteria in your lake reaches a certain level, you can predict that within a certain amount of time, it's highly likely you're going to see a bloom, either surface or subsurface bloom occurring. So the other practical aspect to gathering this information is that we're all concerned about toxins in lakes, and many times the toxin levels are so low as to not be a problem. The other issue is that there's just so many different toxins. Which one should you be testing for? So one of the advantages is this helps you identify when it's time to test for toxins, which toxin you should be testing for. And so in other words, with the fluorometry, you should be able to say, oh, we're reaching a level where I should be concerned about the toxin levels, especially since this is a microcystis lake and the toxin level for microcystins could be very high. So then you would probably go into the next phase, which would be finding a laboratory to test for you. So the world is a lot more complicated than what I've just presented, obviously. We're looking at a simplified view of how this all works. And in fact, we can go from molecules, shown here is essentially the chloroplasts, all the way up to the landscape. And all these parts are interacting, and yet we've simplified it so much. There was an interesting statement by Alfred North Whitehead. He said, seek simplicity, but distrust it. And I believe there's a lot to be gained by thinking about that. In other words, don't fall in love with your own models, even though they may be very seductive. This is a problem, especially whenever we begin to think that the models are reality and find out that, in fact, what is happening isn't what we see in our models. So what do we do about that? Well, we have to, again, adapt. We have to adapt our monitoring techniques to include some of the reality that's needed. Keep it simple, but also complicated enough that it is useful. So we call this sort of our advanced cyanobacteria monitoring programs. And we've got several that we're running now. One is what we call trophic cascades, or top-down food effects. It shouldn't say food we effects, it should simply say food effects. Impacts of fish on water quality and cyanobacteria, but it could be the impacts of any top-down effect. Top-down simply refers to predation, essentially, impacting ultimately the water quality or the cyanobacteria. So we're essentially testing some of this with a series of herring runs on Cape Cod this summer. And we'll be looking also at
okay, I found this on.
We'll be looking at one or two lakes on Martha's Vineyard. So this is going to be very interesting to see how the fish in a lake can impact then the cyanobacteria in the lake. And ultimately, we're also asking the question, could the fish in the lake affect aerosols in the lake? That's another one. So lake aerosols is something we've been working on for nearly a decade. And we've now are in the process of developing more rigorous field methods, but also adapting lab methods that will allow us to collect water and identify whether the water that you've collected has a high likelihood of giving off toxic aerosols. So we're also looking, and I have a graduate student working at on Cape Cod this summer, Haley Carter, who in fact had her first run on Tuesday, and she's looking to help determine what are the drivers of these toxic aerosols. What are the physical factors as well as the biological factors that influence the aerosols coming off of lakes? And then one of our newest programs is what we call SICM, Satellite Integrated Cyanobacteria Monitoring. Integrated because it's also including food web aspects. And so this is the use of satellite reflections data to track cyanobacteria and also make predictions about the toxins, the growth of the toxins, and through our regressions of toxicity. So this is an exciting program. We have quite a number of people participating this summer in collecting ground truthing data so we can test to see, does it really work? So, you know, I think most of you are aware that whether we're talking about what goes on in Earth or between planets, we're really in the age of connectivity, how everything connects around us. And so it seems that we're moving toward considering more and more of the interactions that occur from molecules to whole ecosystems. And in that sense, lake aerosols is one of the ones that illustrates this connectivity, perhaps better than most, because it's connecting then lakes with other lakes, connects the lakes with the watershed, and also connects, it may be connecting lakes with other continents. So I just wanted to very quickly show you the PD clam that we are doing. Some of you have already seen this, but it's simply a portable device you put out over the water, collects then the air coming off of the lake and captures then both the particulate matter as well as the dissolved toxins. And it's been relatively successful. We've been able to show in this, this is simply a sample that for microcystins throughout the summer, there are various levels, but almost always detectable levels in the air. And this is amount of microcystin per meter cubed of air breathed. And this is in 2 lakes then on Cape Cod. We also have looked at two other toxins, BMAA, the neurotoxin, and also most recently anatoxin A, another neurotoxin, and found both of those also present in these aerosols. So it does raise the question of what are the health risks associated with breathing then these aerosols. At this point, we do not know whether there is any measurable risk, but they're present. That's all we can say. I just wanted to show you something that relates to Maine, and that is, this is some of our earliest work that I did with Amanda Murby. Back in 2013, we did the collections published in 2015. And one of the interesting things is, this is the number of cyanobacteria cells per meter cubed of air. And what you see here is Freeport, Maine. And actually, this is not Freeport, it's South Freeport. This is the first aerosol we did on a marine system. This was in the bay at South Freeport. We'd been asked to come in there because of some suspicion that the air could be causing some of the problems that they were having with health. And at that time, they had nothing to compare it with. This past year, we did some sampling on Martha's Vineyard of the water and found that, in fact, the BMAA content in the water and went up with the salinity. So it makes me want to go back to South Freeport and re-examine some of this with much more sophisticated equipment that we have to measure both in the water and in the air. So in conclusion, I simply wanted to say that, you know, blooms we can see. Aerosols, obviously, we would wonder, is there really anything happening here with these aerosol collectors over this lake? But in fact, what we find is by the results that we're getting, in fact, there's both dissolved and particulate matter coming out of the lake. And so that, if you can imagine, this is what the lake would look like if your eyes could resolve what in fact our chemical tests and our biological tests are showing. I'm not sure you want to know this, but this is how I see the lake now. So thank you very much. And thanks to all these people and a lot of other people who have helped out with this, graduate students, Hilary Snook at the EPA Region 1, and of course, Elijah Stommel with the Dartmouth Hitchcock ALS Clinic, Mark Poquist, who's been a supporter of our work for years, and then all of the other that I couldn't mention here because it would take up as much time as I've taken to give this presentation. So I'd be happy to try to answer any questions you might have.
Oops. Hey, Jim. All right, I'm here. There I am. Thanks so much for all that great information. Really, really helpful and interesting for all of us living on the lake. Let me check. If anyone has any questions, please feel free to put them in the Q&A. Tristan Tabor from LSM, Lake Stewards, Maine, did mention that he put a link in the chat. They have a webinar on June 25th that sort of dovetails nicely with this presentation. He's going to be talking about the Bloom Watch app and the applications for that. So if you are intrigued by what you heard today and want to learn more about another approach, definitely listen in on LSM, register for that webinar next, I guess it's in a couple weeks. So a couple questions people have posted. On your aerosols on the Cape Lakes, what was the approximate ratio of the levels in the water during those samplings versus the aerosols?
Well, that's an interesting point. There's a couple of key issues here. One is that, of course, we started off with the idea or the belief that the more toxin you had in the water, the more you'd have in the air. But in fact, it's not quite that simple. The large bloom-forming cyanobacteria don't appear to contribute a great deal to the aerosols directly. And that's something we're checking this summer again. We're doing some experiments where we're actually enhancing the amount of big bloom-forming cyanobacteria and measuring with and without those present how much aerosol we get. So that's a key issue. However, when blooms occur, they often release toxins into the water. And since those toxins are labile, they can evaporate out of the lake into the air. We think that they're probably secondarily contributing to that. There are other factors, and one of my graduate students who finished in 2019, Kate Langley, actually determined that it was the relative temperature of the air and the water that regulated the rate at which the toxins in the cells emerged. And we're testing that at the Cape this summer. So we're going to be seeing if that's in fact, it is in fact important to know how much toxin is in the water. That certainly is one of the factors. But in addition to that, there are at least one or two other important factors that control it. So it's not just that alone. It won't tell you the aerosols.
Interesting. Somebody would like to know about air circulation or aeration systems in lakes. Will they help or hurt HABs?
Well, you know, when we started doing our aerosol testing, that was one of the first things we did. We assumed that if you bubbled water, you would create more aerosols. It seems logical. You're bursting bubbles into the air. So it was almost disheartening when we ran those first tests, because when we bubbled, and this was in the laboratory, we were conducting these tests, when we had flasks that were being bubbled versus flasks that had no bubbling, the aerosol release was higher where there was no bubbling. So that the bubbling effect was not what was creating these aerosols. And these were aerosols measured both as toxins and as cells actually escaping. Now, only certain cyanobacteria can escape. And as I mentioned in the talk, these are these picocyanobacteria. They're so small that they actually can be carried away by the air. So we find relatively few, and we tried measuring this, of larger ones. There is some, but nothing the size of the BFCs. We get get them into the size range of 8 to 10 micrometers, but nothing as big as the filaments and that sort of thing. So the less than 2 fraction is definitely where most of these toxins are coming from, which makes them have such an important role in the lake. And it's especially important because by and large, they're overlooked. So I think that's why we like to draw attention to this.
Yeah. Okay. I have a question. If anyone else has a question, there's time for a couple of more. But is there any, is there eDNA work that's gone on with that can help with identifying these?
Really important. Yeah, I don't. We tried some preliminary work with that. We were using qPCR approaches to collecting. But what we're doing this summer with part of the aerosol is we're trying to determine using DNA extractions to identify what's in the water and what's in the air, and what part of that water fraction actually gets into the air. But we're using then, it's not the same technique for eDNA, but very similar.
Oh.
So yeah, it's sort of an approach to identify everything that's in our samples, and then what fraction of that do we find on the filters we collect above the lake.
Okay.
So hopefully next year when I give this talk, we'll have an answer to that.
Well,
just kidding.
we'd love to have you back. Hopefully next year you could come in person. We hope that we'll be back.
I remember the days. I remember one of your annual meetings I gave a talk at. It was great.
Well,
Yeah.
good. We're hoping it'll be really successful. And I think people are getting tired of these Zooms, but I appreciate everyone who showed up.
Amen.
Our next we don't have any webinars. This is our last one. I mentioned our wake boat webinar has been postponed, but our next one will be June 19th in the Saturday morning, and we'll have Karen Wilson from USM speaking about lake systems and inputs and outputs and alewives and lots of exciting things about
that. Should be a fun talk. That should be a fun talk. I love Karen's.
Yeah, she's great. And as with all of you professors, it's always fun to hear a professor speak, especially because you answer so many. As I write down questions, you always answer them. Like as soon as I write them down, you answer them. So,
so one of the things I'd like to just say, if I could, Susan, is anyone who's interested in sort of taking this multi pigment approach, just contact us. We're expanding this already. We started on the Cape doing a lot of this work, and now we've moved to Martha's Vineyard. We've got some groups in New Hampshire working on this as well, and I don't think we have anyone in Maine that is taking this approach. Basically, that regression that you saw between the pigments as a prediction of the toxins, our goal is for every region to reestablish that, because we don't know if that's going to hold up for every area.
Right.
So the interesting thing is, would it work on Lake Sebago? Would it work on China Lake, for example? Are those different relationships? And we may find that, in fact, it's pretty general, but at this point, we don't know yet. So we know that it works on the lakes in that same region of Cape Cod, where there are lots of very similar lakes, but whether it applies to other regions. So we're anxious to be able to expand this idea and have people help test it.
That's terrific. Well, we have lots of connections to lake associations, and I was actually, our newsletter, our spring newsletter is very late, but the good thing about it is being late is I would love to put something in the newsletter about this project.
Sure.
So I took some notes, and I might pass it by you, but I'd love to get the sort of a synopsis of this talk out to our lake association members, and then see if folks are interested, if we could, Maine Lakes would love to help facilitate that, making that happen.
It's a pretty small investment for, it can be done with an individual on a lake or with a whole lake association, where the lake association has the ability to buy a fluorometer, and we're talking less than $2,000. If they can do that, then that could be processed. All that except the toxin analysis can be processed at the sort of their own lab. On the other hand, if you don't have it and you just wanted to know about your lake, then you can collect the samples and send them to us.
Okay.
So, you know, we don't want to work in a vacuum. We've experienced a lot of that in the past. You know, every group sort of having their own thing. And I think we need to share. Our system isn't perfect, but it's moving us, I think, in the direction that's going to be useful.
Well, that's great. Well, we will definitely be in touch. And somebody asked for your email address. What's the best way to get you?
Jim.Haney, UNH.UNH.edu.
All right, great. And we haven't had any other questions. So with that, let's definitely keep in touch about this, Jim, for Maine and which lakes, you know, if there's a particular lakes or regions, let's keep in touch about that. We'll put something on our newsletter, and we'd love to have you back in person next year, and maybe there'll be a Maine lake in your sample.
Okay, terrific. Thanks so much. Been great being here. Thank you.
Thank you so much. Thanks, Jim. Goodbye, everybody. Thanks for joining us on this afternoon.